Hi, welcome to the third lecture in the Oculomotor mini module, which is part of the PSY6315 Systems Neuroscience course. So I'm going to talk about this paper entitled Dynamic Ensemble Coding of Saccades in the Monkey Superior Colliculus by Goosens and Opstal. So the paper's introduction is laid out like many good papers have. It's got so overall the paper argues for vector encoding of saccadic targets in the superior colliculus, and the introduction of the paper sort of gives you some background. It describes how what the superior colliculus is, gives you some links to some other papers which have provided information about the colliculus. It states that the paper is addressing the place codes to rate code problem that we looked at in the second lecture, on the first two lectures. It gives some description of earlier models and talks about those and with some citations. It describes a gap in the current understanding, and it then goes on to show how the paper presents some sort of solution to address the shortcomings of other models. And to argue in favour of the vector encoding idea. Okay. So previous models, sort of the main model that this paper kind of argues against is the vector averaging model, which you remember from the Grow paper. And vector averaging says that the superior colliculus only specifies the end point for the saccade. So the actual dynamics of the saccade are not specified in a vector average code. The brainstem instead computes the dynamics. You should take a look at this paper here. It's just down at the bottom here. This one, population coding of saccadic eye movements by Lee, Rohrer, and Sparks. And they present some electrical stimulation results to argue in favour of the vector averaging argument. Okay, and you should be able to find, if you look at papers that cite Lee, Rohrer and Sparks, you should be able to find some counter arguments to their work. Okay. So one of the big problems with vector averaging is that it doesn't account for component stretching. So this is the idea that what you've got is the colliculus is this topographic map of the stimulus, and that map. So it's a piece of tissue which actually is physically arranged like the world, and then that map somehow has to be connected up to other bits of brain tissue which are generating dynamic output for the muscles. So this idea splits up the dynamic part of the output into separate downstream brain tissue in the brainstem, and leaves the colliculus as just being a map, okay, just specifying location. And the problem. So what we would say is that in the case of the eye, because we've got two main degrees of freedom, left, right, up, and down. Then we could say we've got 2 or possibly 4 channels in the brainstem. So one for each of the muscles that you're worrying about. Okay, where we're just ignoring the muscle that rotates the eye like that. Okay, and there is a problem with this. To understand the problem, we have to cast our mind back to the main sequence. So if you look at this graph on the left here, this is showing a well-known graph from Robinson, published in 1964. And this shows that as a saccade gets larger in its eccentricity, its duration increases monotonically, but that there's a nonlinear relationship in the peak velocity and the duration. And if I click on to the next graph. This is a graph taken from a Harrison-Wolpert paper, but it just happens to have a nice print of some data here. The data is in the dots, and this shows the peak velocity of a saccade with respect to the amplitude, and you get this characteristic curve where the peak velocity increases as the amplitude increases, but it sort of tails off and it becomes a nonlinear relationship. And if we consider two saccades, suppose we consider now two saccades, a red one and a blue one. The red one is purely horizontal, it's relatively short. The blue one is oblique and it's a bit longer. And notice that the horizontal component of the oblique saccade is exactly the same size as the pure horizontal saccade. So if we look at this graph here, we realise that the short saccade has a relatively low peak velocity, and if we look at other graphs, we know that, or as part of this sort of related relationship, the duration is short. So we get if we plot the velocity versus time for the horizontal saccade, it increases to a maximum and then decreases back to zero over a relatively short period of time. If we then look at the horizontal and vertical components of the oblique saccade, which is longer, or which is more eccentric, then the velocity, the horizontal and vertical components of the velocity increase to their peak and then drop back to zero over a longer time period. Okay, this graph here tells you that because it's got a larger amplitude, it should have a larger peak velocity, and indeed it would have if you added those components up to get the peak velocity. This is only a cartoon diagram, mind you. But this kind of illustrates the problem, because if you've got a brainstem channel which is driving a muscle to carry out this movement, you need some way of communicating to the brainstem channel whether or not it's performing a purely horizontal saccade or it's performing a horizontal component of exactly the same size but which belongs to an oblique saccade. So somehow the brainstem channel that's driving those motor neurons has to know whether to do this short red kind of dynamics or the longer, slower blue dynamics. And that's the problem that's called component stretching. So if the superior colliculus just specifies an endpoint, the levels of activity in the colliculus should have no direct effect on the dynamics of the eye movement. But there are plenty of studies to say they do, in spite of electrical stimulation results where you can stimulate this colliculus and get a good eye movement regardless of the size of the stimulus. Nevertheless, there is a relationship between activity in colliculus and eye movement dynamics, and you could see if you could find some papers that describe that. And from that idea, you might then say, well, in that case, the colliculus might be driving the dynamics directly. It's not an unreasonable thing to imagine. The authors also explain in the introduction their reason for using blink-perturbed saccades. What they note is that straight saccades have very small variations in their peak velocity. They're pretty stereotyped, and they don't change. They don't vary very much. And then they also note that individual neurons, the output of an individual neuron is noisy. You get quite a lot of variation in their output, and that makes it difficult to find a relationship between neuronal firing in a selected neuron and the dynamics of the movement. So what they say is that using blink-perturbed saccades, where you do a puff of air on the eyeball, which elicits a blink, this increases the variation in saccade dynamics, and that makes a relationship between the firing and the dynamics easier to identify. And that's what they do in this paper. Also in the introduction is the author's main hypothesis, which is that each superior colliculus neuron contributes a site-specific mini-vector to the intended movement. So essentially every time a neuron in the colliculus spikes, it adds a little vector to the intended movement. That's all. Simple as that. And then they say that they hypothesize that the output of the colliculus is linearly summed by the brainstem, and we're going to look at their model for how they think that works. Okay, and these lead to a prediction, and the prediction is that there must be a linear relationship between the cumulative number of spikes that you could record in the superior colliculus and the displacement of the eye along the intended trajectory. So the idea is that if you record the spikes, if the eye's got about halfway to its target, you should have recorded about half of the spikes. Okay. I'm not going to talk about all the methods straight up. I'll talk about some of them as we go along through this lecture. But essentially, the paper deals with rhesus monkeys, and they are trained to fixate on targets with their eyes, and they get rewards for when they do what the researchers are asking them to do. They are trained to fixate on an initial point, and then make a prosaccade to a peripheral target. So it's a really simple, straightforward experimental setup, in that you just get the monkeys to look in one place, and then when a light comes on that they want to look at, they look at that, fixate on the new target or saccade to the new target, and they get a reward for doing that. And then whilst this was happening, this is the non-trivial bit, they were recording spike events using electrodes from 139 saccade-related neurons in the superior colliculus. Okay. Yeah, and as I say, we'll go along as we go along. I'll cover other methods. As regards the blink perturbed paradigm that they use, they generate an air puff, blow a little bit of air in at the eye, and that elicits a blink near the saccade onset, and that mechanically disturbs the eye movement. The eyelid going over the eye moves the eyeball and upsets its movement. Oh yeah, and they show the time course of an example lid movement in one of the figures there. Okay, so here's the first result. Figure 4a is just an example saccade. So this shows one control trial and one perturbation trial. This green path here is the blink perturbed saccade. The blue path is the normal or the control trial, and that's the same in all these diagrams. So this A1 shows the two-dimensional trajectories of the eye movement. This is just like the sort of raw trajectory, and it shows the vector that they then call delta E, which is the straight line from the fixation to the target. This color map is the it's the so-called movement field for the endpoint neuron, which is located at the target, and kind of it's included there to make it makes the graph look nice, I think, as much as anything. Okay, and then in A2 it are shown the raw horizontal and vertical rotations of the eyeball as components. So this is an oblique saccade. As we can see here, it's the fixation and the target. There's a horizontal and a vertical component. So that's the raw eye movements there. You can see that the control trials, the blue ones, are quicker and more direct than the blink perturbed trials, which take longer and are a little bit more variable. And then figure A3 shows the spiking pattern of the neuron that happens to be at the endpoint T. Which is they've obviously got a naming scheme for all of the neurons. This is PJ5202, and this shows that you've got more sort of tightly clustered spikes for the control trial than you get for the blink perturbed trial. And remember, this is the same neuron, just on different trials. When you're looking at the paper and understanding it, or reading it, you need to note that there is a change of coordinates. So in order to help them compare saccades, what they do is they rotate, given wherever the target is, they use a standard formula for rotating the coordinate system so that they, instead of using the coordinates, or the axes h and v, they rotate the coordinates around to these new axes h prime and v prime, so that in the rotated coordinate system all saccades are perfectly horizontal, and any vertical movement is a kind of perturbation away from a dead straight saccade. Okay, so that's what that is, and you can find the maths of that rotation, that coordinate rotation, at this link here. Okay. So that takes us on to figure 4b, and so B1 shows this thing called delta E, and then the cumulative spikes, and compares them and shows that they are similar. So this delta E is movement along that rotated coordinate or rotated axis H prime. Okay, and you can see that the cumulative spikes for the unperturbed saccade increases or builds up much more quickly, just like the movement does, compared with the blink perturbed saccade. So there we are. So it shows that the cumulative spikes in the endpoint neuron, so only in the endpoint neuron in this case, for the example saccade, is related to the movement component along delta E. It's worth noting that this is not a population result, it's just for one neuron, but they do show that several trials for the same saccade endpoint, and you get the same results. Okay. And they always start their spike counting 20 milliseconds before the saccade onset. Okay. In figure 4c, they show that the relationship extends to other saccades. So this figure 4c, I'm sorry to have to keep moving this picture around. I should make it smaller, then it'll kind of fit off the end here. That's better. So figure 4c shows that the cumulative spikes in the endpoint neurons for 25 different saccades is related to the movement component along delta E. So this is 25 different saccades and therefore 25 different endpoint neurons, but in each case you count up the spikes and they're related to the movement along delta E. Okay, so that's cool. None of this says anything about the cause of the perturbation away from delta E. So this is showing results for both perturbed and normal saccades. There's blue and there's green in there. It doesn't actually say anything about the cause of the perturbation away from delta E for the normal saccades or the blink perturbed ones. Okay. So the spike counting says the trajectory is somewhere on a straight line. So what this says is if we've counted 1/4 of the maximum spikes for the given endpoint, then we now know that the trajectory must have arrived somewhere along this magenta line. Okay, it could have arrived along this blink perturbed path, and it could be there. It could have travelled along the unblink perturbed path and arrived there, or it could have actually gone in any other path, as long as all we know is that it's somewhere along this magenta line. And then the same goes for half, if we've counted a half of the spikes, the max spikes of the endpoint neuron, or 3/4, we know something about where the saccade has arrived. So the next result they show is in figure 5, and they count up the cumulative spikes for one neuron again, but they look at different saccades. So no longer is this, are all of these saccades, no longer are we counting up the cumulative spikes at the endpoint saccade. We're accumulating the spikes for the same neuron, but different saccades. So A2 in this case is the same as the previous results in figure 4. That is, this is the preferred saccade, so there's 2, and it's that target is at the preferred saccade location for that neuron. So that's the for that saccade you get the most spikes in that neuron. Okay, about 25 in this case. And then what this shows is that even for saccades for which this neuron isn't the preferred neuron, then you still get this relationship between the displacement of the eye and the number of spikes that you count up. You find also that the result for the normal and the blink perturbed saccades is still the same. So there's something very systematic going on here. Okay, so now we're going to talk about reading the population code. So the authors have shown that different saccades give different cumulative spike counts in this systematic way. And that suggests that each neuron is contributing a mini vector, which are then vector summed to give delta E, and that the colliculus is producing a population code. So they decide to test this by passing the spikes that they measure empirically through the simplest model that they can come up with a simple eye model, and then see what the trajectories look like using that model. Okay, so they present this brainstem and eye model here. I'm going to go through this briefly. So essentially what you have is you have the input to the model, which is the spikes on the superior colliculus. Okay, and the input actually has to be spikes on an even grid of locations across the colliculus. Okay, and you have to reconstruct this from the actual spikes that you measure. Okay, so it was clearly too difficult to get. You couldn't just insert an electrode evenly spaced and always find a cell which was responsive to a saccade. So what they had to do is they had to get as many cells as they could that were responsive to saccades wherever they were, and then convert those measured spikes into an even reconstructed spiking grid. And I'll talk about how they did that in a second. So you've got this mapped activity on the colliculus that you measure. You then pass that into this model, which is a two-channel model. You've got a horizontal channel and a vertical channel. And that represents the sort of the brainstem model, which is this part, and then a sort of engineering, it's called a plant, an eye plant here, which represents the physical eye model. And that then generates as output the horizontal position as a function of time and the vertical position as a function of time. Okay, and that gives you the trajectory. So let's just talk quickly about how they estimated the activity in space and time. So suppose they've got the collicular surface here, and they've measured from these blue locations. So those blue dots represent the saccade-related cells that they got measurements from. U and V is the Cartesian coordinate system for the colliculus. That's how we locate the cell. So every cell here has got a U and a V value. And we want to know what the instantaneous firing rate is at any arbitrary location on the colliculus, so that we can feed it into the model. So how to get that? So what they do is they use something called image resampling, and this is a technique that's used. It's used commonly in converting an image. So suppose you've got an image that's on a hexagonal grid. You've collected image pixels on a hexagonal grid. You can resample it to a Cartesian or a rectangular grid using exactly this method. So the first thing you do is you draw the grid you want onto that surface. So that's the red dotted lines. And then you draw the locations of each of those pixels as red dots. So those red dots are the locations where we want to get a kind of reconstructed firing rate. Okay, so we've discretized space into 0.1 millimeter boxes, and we're also going to, because we're dealing with spikes, we're going to need to do spike counting. So we're going to discretize the time into 1 millisecond bins, and we're going to consider the instantaneous firing rate at each, in those 1 millisecond steps. And what we'll do is we'll count up the spikes in those 1 millisecond time bins. Okay. So what you do is you say that for every spike that occurs in any of the saccade-related cells that you actually measure, you create a 2D Gaussian with the standard deviation of the size of the grid, the red grid that you want. Okay, and you center that Gaussian on the location of the cell that you're interested in. And you can then work out that the instantaneous firing rate at any point, any location I, is related to that Gaussian, which is this exp and all this stuff in here, and the number of spikes from the neuron that you're interested in. And to get the firing rate at position I, you have to put one of these Gaussian hills on every single neuron C, and for location I, you have to add up the contribution from each of the neurons that you've got. So neurons that are very, very far away from position I will have almost zero contribution, and neurons that are very close will have a large contribution. And what you do is you only have to worry about neurons that actually fire in the time bin leading up to time t. Okay, so that's what SC of t is, the number of spikes from neuron C within the time step. X minus this stuff here is, if you've dealt with Gaussians, you'll recognise that as the formula for a Gaussian. Omega squared, omega's the standard deviation of it in this case. And then this row here, this is a factor which sort of deals, it normalizes for the density of the measured neurons that you've got, and that's defined in the paper. It's also got a Gaussian component to it. Okay, so that's the idea. You reconstruct the firing rate at positions I from the measured neurons at positions C. Okay, so back to the brainstem model image here. So in this model here, which is this is a kind of quite an engineering looking model. It's especially engineering-y looking because they use Laplace notation. So this, for example, is their model of the eyeball, and it's a very simple first order model, which is what that Laplace notation denotes. But basically this part of the model says that you take all of the inputs and you have a set of varying weights here. So this looks like the first mechanism that we looked at in the Grow paper. And those varying weights, along with a factor gamma x, are added up together and run through a gain B, and they go into the eyeball model, and that gives you a horizontal movement. And this model has got a number of parameters in it. It's got this factor B, which is a gain. It's got a delay where they feed the output of the summed activity back with a delay to essentially inhibit the activity here, to prevent the eyeball just keeping on going and going forever. So they have to figure out what a suitable value for this delay is. They've got this capital T, which is the time constant of the eye model, the eyeball model. And then this gamma x and gamma y, which just slightly modify how much contribution to the horizontal and vertical channels is going to be made from the colliculus. And to work out the correct values for those parameters, they take data for 9 saccades, so they take the firing rate for 9 saccades and the H and V as a function of time, and they adjust those parameters until the model generates H's and V's which are as close as possible to the measured H and V of t. Okay. And then they present the population code results of sort of trying to reconstruct saccades using the firing. So once they've fixed those brainstem model parameters that I mentioned, they don't change them, but then they prove the model by presenting new firing rates for new saccades, and then they compare the output of the model with the measured saccade trajectories. And so here's figure 6b for a horizontal saccade. And they show maps of the reconstructed firing rate at different times. So that's 20 milliseconds before saccade onset, the activity at saccade onset at 20 milliseconds and 40 milliseconds after. Okay, and then they show also these little time courses of firing rates across the rostrocaudal axis versus time. So you can see you've got this little sort of hill of activity, or hill of activity in time, corresponding to the movement. And the green trace here is their reconstructed trajectory, the blue is the measured trajectory, and they are pretty close. In figure 6c, they show that this works for oblique saccades as well as for horizontal saccades. They show that you've got measured saccades in oblique directions are roughly straight, and the reconstructed saccades are also roughly straight. So the technique works, or the model works for oblique saccades as well as for vertical and horizontal saccades. And so what they claim from this is that the straightness of oblique saccades is encoded in the population code of the superior colliculus, because it's only really the population code which can make these things straight or curvy. Okay, there's nothing in the brainstem model that would try and straighten out or do anything special with the different components of the signal that's sent to the muscles. So the second part of figure 6, figures 6D and E, they show that the result holds for different size saccades. So they show for 5 different size saccades. They show that the reconstruction process works for all of them, with the model providing a good fit to the measured data. They show the displacement with time and the saccade speed with time in those two graphs. So the model fits the experimental data well. And so this means that the main sequence is encoded in the superior colliculus activity. And if you compare the peak velocity versus the saccade amplitude presented in figure 6e, and compare that with a graph that I took from a paper by Harris and Wolpert, which just happens to have empirical data in it, you can see that the fit is good, and the sense of the output that you get matches that empirical relationship between the peak velocity and the amplitude. They say that this gives you can consider these the colliculus to produce a dynamic movement field. So it's the movement field measured in the colliculus is dynamic with time. They said because it was shown that each spike can be considered a mini vector, shown with the linear brainstem model results, they reverse the idea and postulate that the cumulative spikes could be calculated with this following equations. Okay, where we say that if we know the number of spikes per degree as a function of r and phi, or as a function of the saccade specification, then we can, along with how far we've moved along that saccade with time at any given time, we can work out the cumulative number of spikes that must have occurred by that time. And in figure 8 they show that does in fact hold true. So they show that if you take lots of different saccade vectors and you look at the cumulative number of spikes generated by one cell, this one ER0902, then you get lots and lots of different temporal evolution of spike counts. So in some cases that cell produces spikes that grow slowly. In some cases it produces spikes that grow fast. And that's with time and also plotted with respect to the eye displacement. So there's an awful lot of variability there for different saccades. But here's the magic bit. If you apply this formula and plot the number of spikes that you would expect given the displacement that you've got and the location of that neuron on the colliculus, then the number of cumulative spikes that you expect matches up really well with the measured cumulative spikes. So that's very nice. That kind of they've shown it one way by showing that the measured spikes produce the trajectories, and they're kind of inverting it and saying that it holds the other way as well here. And then figure 9 just proves that this also extends to blink perturbation. Okay, I'm not going to spend any time on that. So to summarise the sort of results of this paper, there's a few other things in there which I've sort of skipped over. But the main results here are that the number of spikes in a cell, the number of spikes that a cell fires, gives you information about how far along the saccade vector the eye has travelled. And the conclusion is that the activity in the colliculus is a population code in which each spike is a mini-vector, and the sum of those mini-vectors gives a movement command. We get from this work that straightness of saccades is encoded into the superior colliculus population code, and they proved this using this 2D linear brainstem model. And then we have that the relationship between the number of spikes in any cell at any time is given by the static movement field for the saccade. This all relates nicely to stimulation experiments, where the location of a stimulation gives the saccade destination, but intensity of that stimulation will affect the saccade dynamics. But it raises a question. So somehow, across the colliculus, the number of spikes that are fired is just right. Okay, and there's absolutely no mechanism proposed in this paper. This was not the intention of the paper to provide a mechanism. It simply shows that that's what you get. So somehow it's figuring out to be just right, even when there's a blink. So when the blink mechanism kind of fires off and moves the eyelid, something is ensuring that the number of the spiking in the colliculus slows down to deal with the blink. So the blink suppresses collicular activity, but this activity returns later, and still the number of spikes is just right. And also somewhere else a compensation is applied to overcome the deviation in trajectory caused by the blink. So the SC activity encodes the intended trajectory, not the actual trajectory. So how does this relate to the Grow paper? So you might think about which of the three models identified in the Grow paper does this model most resemble. and then think about whether GROW would point to a problem with this paper's theory. Does this paper in any way get around the issues that are pointed out in that paper? I mean, you might just say that this is really just the first mechanism in GROW, which is a weighted summation. And if you look at the model diagram again for the Goosens and Van Opstal paper, you'll see that that weighted summation is right there in the model. So that you can question about whether this points to gaps in the current understanding, given that GROW outlines a number of problems with this computational process, and yet this paper from Goosens and Van Opstal quite convincingly suggests that those spikes on the colliculus are indeed encoding mini-vectors. So you could think about going about trying to find a work that suggests a mechanism for generating the correct number of spikes in the collicular activity. So I'm going to give you some further reading on the mechanisms of decoding in Superior Colliculus. One way of doing that is you can find papers which cite this paper we've just looked at, Goosens and Van Opstal. And you can look at the paper cited by the paper as well, so you can go in both directions. If you have a look on Google Scholar, if you find this paper on Google Scholar, it will give you who cites the paper, and you can obviously look in the paper for where the paper took its inspiration and data. This is a nice paper, a test of spatial temporal decoding mechanisms that has a pertinent comparison of vector summation, vector averaging, and summation with saturation. There's a review paper by Gandhi and Katnani, 2011 as well. Have a look at some of the Superior Colliculus imaging papers from the Basso lab. In those you see some amazing movies indeed of the activity in colliculus for saccades. That's really nice work. You could consider the fact that the colliculus is we've been treating it as just a map, a kind of a map of cells, and just the cell's location on the map is the only thing that matters. But actually, the colliculus is essentially a 7-layered structure. So there are many layers to the neurons, and there could be something much more complicated going on with information flowing between the layers. So that's kind of interesting to think about. There's a follow-on from this from Goosens and Van Opstal again, 2012 in PLOS Computational Biology. You could have a look at that one. That's quite nice. Kind of develops the story on a bit. And a recent paper is from a couple of years ago, Kazap and Van Opstal. They've got a spiking model also in PLOS Computational Biology. And take a look at that. I haven't actually decided what paper is going to go in the fourth lecture. But I might have a look at something from the Basso lab, but we'll see. Time will tell. Well, not much time. I'll have to do it in a couple of days. Great. Thanks very much, then, for watching, and I hope you found it useful, and I'll see you at the next one.